All right, so welcome everybody. We really appreciate you guys being here today. My name is Edward. I am the president of Chicago Ornithological Society, and welcome to Birds and Bytes, our virtual take on birds and beers, especially seeing how we can't share a beer together in person. We figured we'd do that here and also be able to chat with you guys in our community about some of the most hot topics in birding here today, as you would at a bar with your fellow birders. So again, thank you so much for being here. This is a Chicago Ornithological Society program. We're one of the oldest conservation organizations in the city of Chicago. We've been around for more than 100 years. And you know, cyberspace and the 21st century, we're just obviously the first birding group in Chicago to fully embrace this new technology. And we really appreciate it. If you guys want us to learn about upcoming events, please visit our website, chicagobirder.org, and check out some of the awesome programs that we're doing. But enough about that. Tonight we have two awesome guest speakers who we will be introducing in just a moment, but also part of these programs is a partner brewery that we'd like to highlight. So I'm gonna go ahead and pass it over to Chris Mikulek, who will talk about our featured brewery tonight.
Thanks, Edward. Hi, everybody. Thanks for coming. As part of the Birds and Beers kind of take with our Birds and Bytes, we definitely wanted to highlight some of our partner breweries. All right, Steve Huggins on tour. Anyway, wanted to highlight some of our partner breweries that have worked with us in the past that either have, given us space in their brewery for meetings or donations or sponsorships to some of our fundraisers, particularly for the Labatt Woods fundraisers. On Tour is just one of those breweries. They're located in the west side of Chicago. They are relatively new, but extremely supportive of all of our work and what we've been doing. They are obviously closed, but they do deliveries. They are delivering to the entire city of Chicago right now. So if you are looking to have a touchless delivery, please, bring them up on the website and partake. Again, as a community, the birding community requires partners, and these partners have been very generous to us in the past, and it's our turn to return the favor. And when all of this ends, we'll definitely have to stop by and check out their beers at their facility. With that, let me put I'll put their link, their website in the chat so everyone can take a look. With that, I'll pass it on to Nathan.
Great, thanks, Chris. Thank you all for joining us today. You guys are all in for a very special treat. We have two wonderful people here to present today, working at the Field Museum, Dr. John Bates and Dr. Shannon Hackett. And I'll just give you guys a brief overview of the work they do and some of their background before I hand it over. So John went to school down at the University of Arizona, and obviously great birding down there, and that's where things started. But then ended up heading over to LSU to get a PhD. And if you look at his bio online, long list of publications. He's been very prolific and active in the ornithological community for years. His research is focusing largely on genetic structure in tropical birds and other organisms in order to address how this diversity really evolved and how it continues to evolve as the climates change and humans continue to alter their landscape. So additionally, Dr. Shannon Hackett was born over in British Columbia, and when she found birds, ended up heading across the border to the US and down to LSU, where she has been very prolific as well in the field, numerous publications. They're both curators of birds at the Field Museum, and we're honored to have them. Her work focuses largely on describing patterns of avian diversity from different perspectives across multiple time frames and across taxa and geographic borders. She's interested in describing patterns of genetic diversity within and among populations of tropical birds, specifically birds in the neotropics, Africa, and Madagascar, and then relating those patterns of biogeographic histories to these regions, and with an effort to conserve their biodiversity. So from revolutionizing our understanding of how birds are related to one another, to describing new species to science, and everything in between, you guys are in for a great treat. And with that, I'd like to welcome Dr. John Bates and Dr. Shannon Hackett. And John, take it away.
Thanks, Nathan. I'm going to share my screen here. Get this up. What did I do? All right. Can everybody see my screen? Is it on?
Yep,
Yes.
all good.
Good. All right. Well, I want to thank Edward and Nathan for giving us the opportunity to talk to you guys today. Obviously, like everyone else, we're sitting at home these days and working from home and getting a lot of work done, but this is the place we work. And I was looking at this picture and I'm pretty sure I took this on a spring count day 3 or 4 years ago. And the spring count, of course, is supposedly coming up this weekend, and we'll see how it all goes and how different it'll be. But it is a different world that we're in right now. Being away from the museum, just like everybody else, we're struggling with what that means and trying to figure things out and working on Zoom. And so I figured out today I'd just give you a little bit of background, more along the lines of sort of what people do in the building in birds and where we've been going. And I wanted to start, Nathan already gave an introduction about where I come from, but I grew up in Tucson, Arizona, and I got interested in birds actually because I come from a birding family. This is me giving a talk in the museum, but the photo is of me in a boat in 1971, and that's my dad and my brother in the front, and I'm the one in the middle. And we're in the Caroni Swamp in Trinidad, getting ready to go out to see the evening return of scarlet ibis to the mangroves there. So in addition to growing up in Tucson, which is, as you know, an awesome place for birds, I also grew up in a family where we designed family trips around trying to go to interesting places like Colombia and Peru and Africa over the course of my lifetime. And so that was an incredible introduction. And then fast forward to 1984, and I had just finished my undergraduate degree at the University of Arizona, and Doug Stotts, of all people, who had ties to Tucson too, was back in Arizona after having spent 6 months in Brazil. And he walked into my major professor, Steve Russell's office, and he said, "Steve, they're looking for people to volunteer as bird banders down in Manaus on this minimum critical size of ecosystems project. Do you know of anybody?" And Steve said, "Yeah, there's, John Bates, I think, is working in a greenhouse." And I heard about this project, and it was December, and 2 weeks later, I showed up in Manaus, Brazil, not knowing anybody, and proceeded to spend 6 months banding birds in Manaus, and being in a situation where I was in the field with materos like Valchino here, who didn't speak any English. And it was just an incredible experience that introduced me not only to the tropics, but also to doing science in the tropics, because the goal of this project was to look at the effects of forest fragmentation by gathering data before the fragments were made and then following the fragments after they were made. And so it was a great introduction to the tropics. And I came back and I knew I wanted to continue on in ornithology at that point. And so I went to graduate school and got a master's degree at the University of Arizona, where I studied the winter ecology of gray vireos. And this is Puerto Lobos, which is on the Gulf of California. And my two study sites were straight ahead of you there in the hills to the south and then to the north. And, this is where I was studying ecology. Gray vireo is a tremendous bird. They eat fruit in the winter and they defend these individual territories. And it was a lot of fun, but I also got introduced to evolution during this time. And at that point, I realized that I thought that was something that I wanted to do for the rest of my career. So as I applied to graduate school, as Nathan said, I came close to going to Wayne State University, and at the last minute, LSU called, and I ended up going to LSU. And what I was able to do at LSU, again, was by luck, get into a place where I discovered an ecosystem where we were doing inventory work in northeastern Santa Cruz, Bolivia, a place called the Serranía de Huanchaca in Nolquem Mercado National Park. And I was able to design a dissertation that built on my interest in evolutionary biology, looking at the effects of forest fragmentation by getting samples and different forest fragments on top of this plateau and comparing the genetic structure in those forest fragments to continuous forest sites below the plateau. And, you know, so that was a great thing. That's where I met Shannon. And Shannon went off to a postdoc at the American Museum, and I followed her. And were married by that point. And then in 1995, we ended up coming to Chicago. And we joined the Field Museum, which has this incredible history in ornithology, 126 years old, where you have people, mostly white, not mostly, all white men in the past, working all over the world, collecting birds and bringing back to the collection. So you've got George Cherry here sitting in a chair in Mongolia, Austin Rand, Ashley Hind, and Charles Conover, one of the first curators of the museum. And so, you know, I always like to say that we stand on the shoulder of giants. And one of my all-time favorite people is Mel Traylor, who's born and bred in the north suburbs of Chicago and had an association with the Field Museum that lasted for 70 years. And, you know, here he is paired up against Shannon in a picture that we both love, but we also like to point out that the reason all these birds are out was actually that an air conditioner in the museum had broken and a bunch of water had flowed all over the game bird cases. And so they weren't really wet, but we were drying them out to get them ready to go back in when this picture was taken. And I just want to point out that this is one of the things that's happening right now is that, you know, we've got a skeleton crew at the museum, but the dedicated people on the collection staff are walking around the collection areas on a regular basis to keep track of what's going on and making sure that any of the rains that we have don't cause damage in the collections or anywhere else in the building. You know, I said that when we got to the Field Museum, almost all the previous people associated had been white males, and that's true. And one of the things that's been most rewarding, I think, over the course of our careers is watching the face of the people doing this research change in lots of different ways. And so these are just a few of our former students, and they come up from this spectrum of the community that's interested in doing research on birds. So there are people like Ben Winger and Nick Block who are hardcore birders, and there are people like Jason Weckstein and Pepe Teo, and Pepe's a Peruvian, Isabel Caballero is Argentinian, Ana Carnaval is Brazilian. And, you know, this has been one of the greatest things that is out there that I worry about going forward. We have to maintain is bringing together people from all over the world to answer questions about birds like we've been able to do. There's an incredible network out there. These folks have gone on to be professors at local universities and colleges, curators of birds at various institutions. And so, you know, this is a tradition that needs to get bigger if we're going to effectively conserve and study the birds around the world the way we should be. So the best thing about what we do, as far as I'm concerned, is actually get to the field. And of course, that's not happening now either. Some of our graduate students had plans to go to Ecuador this year. Those are probably put on hold. We've got a funded trip to Antarctica that we hope will happen in January of 2021, and we'll just have to see. But you know, this is the most amazing thing, is to get to some of these places. So the picture on the top is the north tip of Madagascar. The picture in the middle is a camp in Malawi. And the picture on the bottom is a boat that we spent a month and a half on going up the Rio Japurá River in the western part of Brazil. And again, all these trips are designed to do the best research we possibly can. And we don't do it in the same way that we did historically. You know, in the past, specimens were gone out, that were permissions were granted, specimens were gathered. There was no place to house them in most of the countries where people were working, so they all came back to the museum. And that's all changed now. I mean, when we go to the field, we're always working with locals. We're often able to work with local institutions that have collections, and we're building and training those institutions to do their own types of research. And I put this slide in because it says at the top, access and benefit sharing. And this is an interesting thing. The US is the only institution, the only country in the world that has not signed the Convention on Biological Diversity. And one of the discussions that's coming out of that is that the world is divided up in the eyes of many people associated with the convention into countries that are providers and countries that are users. And countries like the United States and countries in Europe are considered to be users. And a lot of this terminology, which is something I'm involved in discussions about lately, centers around the idea of access and benefit sharing. And what I'd like to point out is that this is something that we as an institution have been trying to improve upon and do better all along. So in these pictures here, you've got people working in the labs or the collections who come from Mexico, Ivory Coast, Kenya, Rwanda, and Uganda. And so, you know, one of the things we're really trying to do is build the next generation of scientists, not just in the US, but in other parts of the world. And of course, that extends out into the field work we do. As I said, we're always bringing people into the field with us. So I'm sitting there on the upper left-hand picture with colleagues in the Eastern Democratic Republic of Congo. And in the right-hand picture on the top is a camp in the southern edge of Amazonia in Brazil. And then in the bottom is a picture from Cordillera Azul National Park in the foothills of the Eastern Andes. And you know, there are people in this picture, such as the guy that I'm sitting next to in the Congo photographed, who's now been trained he got his bachelor's and master's degree with, and then PhD with support that we were able to leverage over the course of our time here in Chicago. And he's now the rector of the largest teacher training university in the Eastern DRC. And Luis Queto, I was just emailing with him the last couple of days. He's the Peruvian with his arms crossed in the picture from Cordillera Azul, is in graduate school getting his PhD at the University of Oklahoma right now. So all these efforts are trying, you know, we're trying to move forward the capacity for doing things. And we were talking about some of the ways in which we can communicate with people now in a situation where they can't come into the museum and we can't go into the collections. And, you know, one of the things we've done is put our data out online. So you know, we have a collection of over 500,000 specimens, and you can access that database from a number of portals online and get information about the collection. I also wanted to emphasize that one of the things that always comes up is, why do you need to collect birds? Or anything else these days. And one of the things I would argue is that we're doing this in ways that we never dreamed of, even when we started as graduate students. And so we've developed this concept in the community that's called the extended specimen. And so you may see a specimen in a tray with a label on it. That label will have detailed geographic information and things, but there'll also be tissues, there may be parasites, there'll be stomach contents, there can be swabs for various pathogens. There's all kinds of information that's coming from these specimens going into databases, and there it's there for future users. And so this is it's an incredible time period for us with respect to what's possible with respect in terms of research. And I just threw this slide in because Shannon spearheaded a number of research projects in the collection based on doing survey work for parasites and pathogens. And we're now where we are tonight because of a virus that probably crossed over from pangolins and bats, potentially. We don't know for sure, in China. But you know, one of the things we would point out is that the solution to figuring out what's going on with respect to these events actually lies in understanding the hosts that these are actually crossing over from, and as much as we can about them. And we actually have tools now to do that in terms of the genomic sequencing we can do. But oftentimes what we don't have is actually the sampling from the hosts, and in order to understand what populations of the hosts do and don't harbor the virus and which ones are affected or not by the virus. And so I can't emphasize more that collections can play a huge role down the road with respect to getting ahead of things so that viruses don't end up spreading the way they do. Nathan mentioned new species. Sometimes the new species aren't birds. They can be other things if you're collecting for them. So we've been collecting endoparasites on our trips, and Kaylin Patitucci did her master's degree at the University of North Dakota with a colleague of ours. And she described recently 2 new species of digenarians, digenians. And these are little microorganisms, and sometimes one of the rewards you get for doing this kind of thing is, you notice the one on the right is named Avula forbatesi. So I got a digenian named after me. On the left, the digenian is named after Tatiana Pequeña, who's the Peruvian collaborator that helped us get permission to work in Cordillera Azul National Park, where this green and rufous kingfisher was found. But what I want to emphasize is, here's a new species, 2 new species that basically are known from one specimen of kingfisher in the foothills of the Amazon basin. We know nothing about the distributions or the biology of those 2 new species. And so there's so much more we need to learn about biodiversity. And again, technology is helping us out in ways that we never even dreamed of. We're involved in an NSF supported project called OVERT, which is designed to CT scan vertebrates and make those CT scans generally available for researchers. So we've been taking our fluid preserved specimens from the collections down to the University of Chicago and CT scanning those in the lab of a colleague of ours. And then those get put into a public data repository where anybody can access them. And there's all kinds of things that can be learned about the internal structure and function of vertebrates via CT scanning that we just haven't had access to previously. Our students, while they can't go into the field and are still going out and publishing papers, I wanted to mention a couple of them that just came out. One is by Valentina Gomez, who's a graduate student at the University of Illinois Chicago studying migration in neotropical flycatchers. And she just got a paper in Current Biology, which is a beautiful study of how migration is lost across the tree and what that might mean in terms of speciation. And then Shane Dubé, who graduated a couple of years ago now, just published one of his dissertation chapters in Journal of Animal Ecology. And this was a study of the physiology surrounding differential patterns of migration in 3 closely related members of bush robins in the genus Tarsiger. And so these bush robins come back at different times into these extremely high elevation breeding areas in the Himalayas. And Shane was able to show that they have very different patterns in terms of their muscle structure. We've also got some neat projects going on with migration in North American birds. And so Heather Skeen is working on a fascinating project with Kirtland's warblers, where she's actually able to get fecal samples of birds, the same individual birds, because of tracking on their wintering grounds, when they arrive in Michigan on their breeding grounds, and then later in the season. And so these are all these projects that we could never have dreamed about when we first started. And of course, many of you will know most of the people in these pictures. So Dave Willard, Tom Noske, Ben Marks, and Mary Hennon have been the long-term centerpieces of the collection management team at the Field Museum. And they've been going on and studying migration. And of course, that developed into the Chicago collision monitors. And there may be some people on this listening in tonight who are in this picture. And it's a team that's actually still going strong here in 2020 and going out and surveying what's going on there. And that's resulted in these incredible data sets that you see. So this is a paper that just came out on the changes in wing length that appear to be consistent with patterns of global warming across these birds. And again, I can come back to this COVID example and say where if you're watching the news today, it's all about sample size. And one of the things that we have as the result of Dave Willard's incredible work measuring all these birds through the years is incredible sample sizes for the common migrants that show these patterns very strongly, that these birds are getting shorter, smaller body size. Their wings are actually not changing or getting longer. And so this is a pattern that's consistent with climate change. So I'm going to turn it over to Shannon now. And she'll talk to you about some of the things that she's been doing.
Shannon, you're muted.
So, like John said, we came to the museum together right at the very end of 1995. I had a baby shortly thereafter. We got here, and 10 days after our son was born in late 1996, John went to the field in Bolivia for a couple of weeks. So that's kind of the life of a ornithology curator. So I grew up in the top left picture of this slide in the mountains of British Columbia. It's actually my background image comes from my family's farm there. And this little town, the biggest things to happen there are hockey and baseball. And there's a whole bunch of very famous athletes that have come from this little town. But, you know, I was the weird girl who really liked to be outside. I did not wear Easter bonnets and nice shoes like the girls in my community were supposed to. I wanted to play baseball and hockey with the boys, and I was told girls don't do that. I never from my parents though. I was the first girl in my high school to take physics. So, you know, I always feel like I just never quite fit. And that's why I think it's so great for kids these days that the internet gives them access to people who are like them. So you don't have to be wandering around like me thinking, am I the only person like me in the whole world? Am I the only person who keeps spiders in my mom's costume jewelry boxes in the bedroom? Which that actually might still be true. But my entree to the rest of the world, John traveled. I did not grow up knowing anything about birds. And so, you know, I think so many, especially young boys, get into ornithology through bird watching, which is a relatively solo event, but fairly heavily male. And you know, there are like the feminist bird clubs and things like that now that are opening it up a bit. But my entree to the world from this tiny town, so there's 7,000 people in the town that I grew up in, and my entree was through books. My entree to everything was through books, whether it was my dad's war books, because I really got into warplanes, whether it was Harlequin romances that I read by the tens of thousands because my parents couldn't afford to buy me books, or books that in the library, like this World of Birds book. And so that's how I interacted with the world when I was a little kid. I went to the University of Victoria on Vancouver Island. I never even thought of applying to more schools. When I see what happens to my son and his friends when it comes to going into universities, I think that things are kind of crazy. I was too afraid to apply to the University of British Columbia because it was too big. So I ended up going to the University of Victoria, the one and only school I applied to, and I got a Canadian government scholarship to where $1,400 of those scholarships paid for almost my entire tuition for the 4 years I was at university. Times are really different now. I always say that when our son is in school, and that it's the equivalent of buying a Mercedes every year that you don't drive. for him to go to school. And from there, I was going to be a medical doctor. I actually got all the way through being an undergraduate without knowing how people became professors. In fact, I didn't know anybody who had an undergraduate degree even when I was growing up. I always tell people I couldn't have been me growing up because I didn't know I existed. I never went to a museum until I went away to university. I probably knew the definition of the word curator. I might have had to look it up in a dictionary, but I certainly had no lived experience inside of a museum. And I didn't know anything about birds, but I was just really curious about biology. So the only career counseling I got was, you're too smart to be a nurse, you should be a doctor, and you should stop being so weird. Almost no girls, actually almost no one, but certainly there were 4 girls in my high school class who went away to university. 2 of them became nurses, 1 became a teacher, and then there was me. And so I ended up getting a degree in biology without ever going to a single office hour, without saying hello to a professor except hello in the hall. Never went to an office hour, never talked to them. I didn't know anything about independent research. I knew we called people doctors, but I had no clue why. I don't even remember what I thought at that time about, I was just very polite. So I knew you called them doctor, so that's what I did. And it wasn't until I had a summer job working for the Royal British Columbia Museum, which if you've ever been to Victoria, I encourage you to go to. It's a spectacular place, where I was lucky enough to find a mentor who helped me understand what, what my options were. He told me one day, you're just wasted going to medical school. You need to be a research scientist. And he helped me understand what that meant. And then I took the GREs 5 days later, blind, without studying, not having known a single thing about them. I applied to work with Bob Zink without knowing a single thing about him, but except that at the time he was in California, and I thought, okay, I can handle California, only to learn that he was actually in Louisiana, which when I showed my parents where that was on the map, they're like, holy cow. And then, 2 months later, I was in an unair conditioned car by myself, driving across the entire country to Louisiana. And it was when I got there, despite the heat and humidity, that I realized there was a place where I could fit, where my incessant curiosity that had just exhausted people my whole life was actually a virtue. You were supposed to question, you were supposed to learn deeply and interact intellectually over everything you read. And it's fantastic. And I'm still like that to this day. John can tell you that I never stop bugging people. I never stop asking questions, whether that's COVID-19 to hockey, to whatever. It's constant learning. But that mentorship, that like random association of mentorship, became something that I think is a guiding principle in my life that matters to me. I didn't understand how much it would matter to me, but certainly having a child made it matter to me more than almost anything. So then we went to the New York City, where there were more people on the block that John and I lived in than in the whole town I grew up in, and which was really amazing. And then we came to the museum, and when I look at that picture of the Field Museum on the right, and I see all those people in there, and I wonder when the next time it's actually going to look like that because now it's really freaky when you go inside the museum, because you're in there by yourself and it's completely empty. John and I have been in there once, and it was really a bit weird. So one of the things I did was create a DNA residency program when we opened the DNA Discovery Center. So if you come to the museum, we have a glass-fronted lab, which was one of the great things about my job, is that I could kind of dream things into existence. And the museum's small enough that you can kind of make things happen if all the circumstances align properly. And so when we opened the DNA Discovery Center, I wanted to have a high school internship program, because I didn't want any little girl to ever be told again, you're too smart to be a nurse, you should be a doctor, as if those are the only careers you can have if you're interested in biology. But you can't be something you don't know. And it's our obligation to tell people about careers that they don't know. So this is a high school program where high school students and high school biology teachers form teams and work on real research projects in our lab. And that leads us to people like Nathan, who spent a significant amount of time with us through the years, and as most of you who know him know of his personality, but I have to show this. And this is actually not that far away from a representative day. So you think museums and science is kind of staid and boring, but we're not. Okay, well, that's a little fun at Nathan's expense. You know, Nathan can tell you what it's like behind the scenes. We actually do love each other, and it is a lot of fun. And science is not a solo endeavor done by white males, middle-aged or older white males. Science is a collaborative experience, and everything in my career has done that. And one of the things that I've been grateful for, which is to kind of throw off some of the trappings of what an academic is supposed to be, and actually enjoy interacting with people and teaching people and showing people the collections. And it's some of the funnest things we do. And it's one of the weirdest things about what we're doing tonight, which is normally we would both, John and I would be there, and we'd have trays of birds. It's kind of like a security blanket, right? Because if you think we're boring, we can at least show you specimens, and then you'll think the specimens are cool, and then maybe we're cool by, you know, by being adjacent to the specimens. So it's really weird to give presentations without actual specimens. So I'm going to talk about trees of life. And so this is my family. You guys are all, I'm going to get into detail about what a tree of life is for a couple seconds. This is my family on the golf course in British Columbia. So my mom is in this picture, John and me, my brother, our kids are all in this picture. And this is John's family. And I think there are over 30 million hits if you do genealogy in Google. So that's how many different places you could go to on the web through genealogies. And so this is a tree of life, and you may have done this yourself using, there's lots of tools on the web that will let you create one, but I created this really brief one. So that's John's mom at the top. John has 2 siblings. Each one of them is married, and we all have kids at this point. So this is a tree of life. So we are connected through ancestors and descendants through the molecule of DNA by this same process. And okay, so now I'm about to get really sappy and weird, but it's that process of ancestry and descent that I think is the mystery of life. You are connected to everything that's ever lived on Earth by this exact same process, through DNA, through ancestors and descendants. And that connects you to bacteria. It connects you to everything that's ever lived, extant and extinct. So you can make trees of life in varying ways. You can make them from single genes, like on the left side of this screen. That's like, I think that's over almost 6,000 individual different species from an RNA gene that is found throughout life. Or you can make genes based on whole genomes. So the genome is the sum total of the DNA that any organism have. And you can make trees based on genomes. That's a bacterial tree of life that is best based on whole genomes of bacteria. And this is a poster actually, but I really love the way it kind of connects everything. So I'm going to talk about birds in this tree of life. So we're going to go to this part of the tree. And some of you may have heard this again, but this is what Nathan wanted to hear more of. But about 10 years ago, this is what we knew about the tree of life for birds. So that is a comb. Trees, you know, have branching structures. Trees don't look like combs, at least not well-resolved trees. And right, 10 years ago, we knew virtually nothing about how the major lineages of birds, like how the orders and families of birds were related to each other. And we knew just a few things. We knew the ratites, the large flightless things, were at the base of the tree. We knew that ducks and chickens came after that. And then we have this big radiation, which encompasses almost all birds that we called neo-aves. And within neo-aves, we knew very little about the relationships among these lineages. And at that point, virtually every permutation and combination of relationships had been suggested. So what I want to talk to you about as part of this project, which is to turn that bush on the left, that comb on the left, into a resolved tree of life on the right. And we did this through more genomic approaches with about 260 species, give or take. And we had a fairly well-resolved tree that had a lot of mind-bending sets of relationships that people, including my postdoctoral advisor, one of them, said was crazy, that we were crazy. And who did we think we are anyways? And this tree is crazy. And I'll show you a little bit of its craziness here. So that tree, which has a lot of branching points, and one of the great things about working in the museum is that you work on teams with people to make complex scientific issues into formats and approaches that can reach larger groups of people. And so we took this really complicated tree on the right, turned it into a simplified tree to look at taxonomy. And John can tell you I have a lot of problems with taxonomy. When I said this last night before we were, when we were practicing, making sure we knew how to share our screens, I think there was a little bit of horror on the part of some people because taxonomy can be so political. So I'm not going to spend too much time talking about taxonomy. I think trees speak for themselves. I know taxonomies have usefulness, but they also have a political nature, and I don't feel very comfortable with this. But one of the things our tree showed really dramatically was that 35% of the orders of birds were not monophyletic. So they weren't all each other's closest relatives. So what that means is when you open your field guide, most people in the ornithological community and in the birdwatching community thought that when they opened their field guide, that field guide told them something about the relationships of birds. So you would find loons near grebes, you know, water birds. It kind of makes sense. But the reality is that a large percentage of those relationships are not actually borne out by any data. And as soon as we put critical eyes to these things, a lot of the things fell apart. So if you expect your field guide to reflect the tree of life for birds, there needed to be a whole bunch of changes, which of course for people like us whose collections are organized taxonomically makes you shake your head and have like fits of nightmares of horror about how you could ever encompass a complicated tree that changed almost overnight so dramatically you can't even imagine. How do you do that? Well, we didn't change our collections. So it's about telling people how to get to where they want to go in the collections. And our exhibitions people turned it into that complicated tree into something that is in our exhibitions area. So if you go into the bird halls in the Field Museum, you'll see this tree, which is very approachable for people to understand complex patterns, almost like, you know, Lego style representations. We're going to take a trip through some of these things. We're going to start here at the ostriches, large flightless birds. We can't travel the world, but we can talk about birds from all over the world. And so this is one of the things we've known for quite a while, that ostriches, Rheas, I have never seen, well, I've seen ostriches, but you know, a lot of these birds are mysterious. To me, I've seen them by tissue, by DNA, and by specimens, but a lot of them not live. And so one of the first things, this is probably the thing that happened that people said was absolutely the craziest thing. So we got a tree of life for these birds. And what we found was that, I should have turned my thing into a pointer. What we found was that the flighted birds, so these Tinamous in the bottom right, are the only members of this lineage that fly. And historically, they were thought to be outside of the relationships of ostriches, cassowaries, emus, Rheas, but we found them nested within. Tinamous were nested within as the sister group of Rheas. And everybody said that can't be. It's not possible, because we all knew that these large things were united by flightlessness. But it turns out that that's not the case. That that we think, so you could have hypothesized that the flight was lost in the ancestor of ratites and then regained in Tinamous. That's what the tree would suggest, but that's actually probably not very likely, because independent flight has not originated very many times in the evolutionary history of life on Earth. So it's probably more likely that flight was lost independently in each of these large continental flightless groups of birds, and that Tinamous retained the primitive ancestral condition of flight. And after we were called crazy for this relationship, it turns out people tried really hard to make it not so. And there were more genomic approaches and more sophisticated approaches to this. And virtually all of them showed the exact same thing, that Tinamous are indeed nested within the flightless birds. And so that was one of the dominoes to fall. So this is like the falling of long held true things that we thought of as truths, because there's nothing like data to get in the way of your preconceived notions of the way the world is supposed to work. And these data really got in the way of the preconceived notions about how bird and their evolution was supposed to work. So that's what happened. Like immediately that became the case. So where are we going next? Oh, we're going to, this is one of my favorite birds. So this is a shoebill. When first time I saw this bird in a picture book, I'm like, what the heck is that? And then you see the specimen up live. That thing is over 3 feet tall. And it is the weirdest looking. It looks prehistoric. If you look at that and you think that has to be like an artist rendition of some prehistoric thing that walked around the time of dinosaurs. But shoebills have had a very tortured taxonomic history. They've been placed in a number of different orders. And the thing is really cool anyways. There are videos you can see online. I've never seen a live one, but one of my favorite people, Holly Lutz, has these great video of it eating a lungfish in Africa. These things are lungfish specialists. So you look at that bird, you're like, what the heck is it? So is it a stork? Is it not? Is it by itself? Is it not? What is it? It turns out that when you use DNA data, shoebills are closely related to pelicans and hammerkops. So pelicans are more closely related to this shoebill than they are to other things that have been in Pelecaniformes. So in other words, Pelecaniformes, as you thought of it 10 years ago, does not exist, not unless you put in shoebills and hammerkops. And this was extremely strongly related. In fact, Pelecaniformes has got some really weird things, and Gruiformes is like nonsensical even to this day. We don't really know what some of these orders of birds actually are, but the DNA told us things that the morphology was having trouble telling us, because a lot of bird groups look really unique, but the idea of what they share inside can be very complicated. So that was another really great result from this study. Oh yeah, now we're going to go to hummingbirds. John and I were looking at hummingbird pictures today because I have had this slide for a while, but I wanted some new hummingbirds in it. So another really bizarre thing that people said was absolutely crazy and nuts from this study was showing that that we had some orders that were nested within other orders. And so for a long time, we've known that hummingbirds and swifts were each other's closest relatives, but we didn't know what they were related to. And DNA tells us pretty clearly that they're nested within nightjars. And so whoever saw that, what did someone have a whippoorwill? Did they have in their yard in that chat at the beginning? A jealous bird for me making my blue jay seem way less than. But in this case, we found very clear evidence that hummingbirds and swifts were nested within the Caprimulgiformes. And the sister taxon of owlet nightjars, again, something I've only seen specimens of and never seen in real life, are really strongly supported relationships, which is really an interesting thing. if you're going to study the evolution of avian vision, for example. So what happens to a group of birds that are diurnal, active during the day, if they originate from a group of birds that are largely nocturnal? What does that do to the evolution? And when you know this tree, it lets you ask that question in better ways than you could before. So yeah, orders within orders is really not supposed to do that. Oh, and now we're going to probably the thing that was like the most controversial of all of the controversial findings, which is songbirds. And so, you know, songbirds, the largest group of birds, half of the birds of the world are songbirds, with all this incredible song learning, display behavior, morphological variation, ecological variation, niche variation, really incredible. But we didn't know the context that songbirds originated from. And I remember the first time we got results from this, I looked at the tree and I'm like, no way. I'm like, we got to do it again. There's something wrong. We have to try again. We have to do the analysis again. And I remember it was right before Christmas that we got this results out of the computer after waiting. Well, first off, we waited for a really long time. Then we got a result that was crazy because someone had made a cut and paste error in a data matrix, which caused me and Sushma Reddy, who was working with me, who's now at the University of Minnesota, to sob in her office for like 2 hours right before Christmas. And we fixed the cut and paste error and started the computer again, because when you get these large data sets, it's not, you don't just like click a button and then get a result. You click a button and then you check it obsessively for sometimes months before you get an actual result to come out, depending on what you're doing. But these days it's a bit faster, but it still takes weeks to get real results. And so we did it again, and what we found was that songbirds are related to parrots. So parrots are the closest living relatives of songbirds. And the next branch out of that are falcons. So yes, indeed, falcons are more closely related to cardinals than they are to red-tailed hawks. And that was thought of as very heretical. And there was a lot of work to try to understand how that could possibly be, but every subsequent study that has examined enough genes and with enough taxa has found the exact same thing, that falcons do not go with the rest of Falconiformes. So you can start to see just how many changes to taxonomies have been wrought over the last 10 years. There's a lot. The taxonomy that you saw 10 years ago bears no resemblance to most of the modern taxonomies that you might look at. If you go look at Clements, for example, online, it's nice. You can download an Excel spreadsheet that has all of the taxonomic things. And if you compare, and they have versions, so you can go back and see older versions of Clements just to see how dramatic the taxonomic changes are. And this result is really, really amazing. Not just because it was so unexpected, but because it really gets at something fundamental about songbirds, which is the evolution of song learning. So what does it mean if the closest living relative of songbirds are parrots that also are famous for learning their songs? And that opens all new avenues of research that people are now taking. So this tree, and it opens up science in a way that nothing else could have. And that is, so now we're looking, well, do songbirds and parrots learn songs in the same way? Are their underlying brains the same? Neurological pathways, are they the same? Genetics, the genetics of song, of vocal learning, are those the same between parrots and songbirds? And so the only way you can answer that question is because you have the phylogeny, you have the tree of life that tells you that. And then the other thing you learn is that being a raptor, having a raptorial body, is primitive. It is not a shared characteristic. So it does not define groups like Falconiformes. Instead, we think it's a primitive morphological, ecological type at the base of what we call the landbird clade of birds. And that again gives you the opportunity to look at what changed between passerines parrots and falcons, genetically, behaviorally, genomically. And then my life would not be complete if I didn't tell this to you. If there's anybody who's either a high school teacher or looks at high school textbooks, I'm going to tell you that virtually everything in every textbook except for one in the US is wrong when it comes to Darwin's finches. Like 100% flat out wrong. It's been wrong for over 10 years and it still doesn't change. This is like the most trailingest aspects of textbooks. They're recalcitrant to talking about the real picture of Darwin's finches. So I've, again, never seen a live Darwin's finch. I've measured a lot of dead Darwin's finches. And Darwin's finches are incredibly complicated. We've known this for forever. In fact, Darwin thought Darwin's finches were so complicated that he gave them to John Gould to look at and paint, and he bailed on them and gave them to his wife Elizabeth. And so most of the paintings of Darwin's finches are actually done by him, by her, I mean, and not even him. So Darwin's finches, the most famous group of birds in the world, the birds that spawned our understanding of evolutionary biology. I've never been to the Galapagos, but I think it's a fantastic thing to do. I was offered a time I could have gone on a trip to the Galapagos. Our son was like 8 or 9 years old at that time, and I don't remember where John was, but he wasn't home for the summer, which was pretty common. And the president of the museum said, well, you're going to lead the tour. Don't you want to go? And I'm like, well, of course I want to go. Can I bring my son? And he just looked at me. And I'm like, well, you know, I am the mother. He's not a cat. I can't just leave out a whole bunch of food for 2 weeks, a bunch of water, and then take off and hope, you know, that's my biological fitness. Biologists aren't going to leave their fitness like that. But anyway, so I didn't get to go to the Galapagos. Other people did. But I can study Darwin's finches. And so this is the most studied group of New World birds. I'm going to tell you, and we're not going to talk about it, but nobody, in my opinion, nobody knows what a species of Darwin's finches are. And I don't know how many there are. I think there might be only a few. And so, yeah. So it turns out that when you look at DNA, Darwin's finches actually aren't finches, they're tanagers. So that's the take-home message. So the whole wayward story you think about Darwin's finches, like some sparrows being blown out over the ocean, landing on the Galapagos, and then diversifying, is not true, or it's not complete minimally. So it turns out that Darwin's finches are tanagers. Not only are they tanagers, they're incredibly closely related to a group of birds who have also had fairly tortured taxonomic histories that were never thought to be tanagers before. And that includes Puerto Rican bullfinches and banana quits, orange quits, and things like that. So from now on, every time you hear someone call them Darwin's finches, you should think in your head that they're Darwin's tanagers. And that matters because the evolution of beaks, which have been such at the foundation of our understanding of Darwin's finches, can't be understood if you don't understand that the source of Darwin's finches involves trips through the Caribbean with birds that have almost as much beak morphology variation as they do. So you can go and amaze your friends if you could see them personally, but otherwise in 2 dimensions, you can tell them, you can tell your kids that their biology textbook's almost certainly wrong.
Hey, Shannon, I think if it's all right at this point, I think we probably got a bunch of questions. So
yep. So I just wanted to show the pictures of Darwin's finches.
Absolutely fascinating.
So thanks for listening, everyone, and hopefully you can come back to the museum, or don't hesitate to email John and I if you want to know more. Can't promise you tours, but we can talk to you.
Great. So
I'm going to stop sharing my.
So I saw that we had some questions in here. The first one that, well, first off, I should thank you both for both very incredible presentations. I'm sure everyone's learning plenty. And so, yeah, if everybody, there's a chat on the side. If you want to dump your questions there, we'll highlight various questions. The first one that I noticed was from Alyssa Schoenberger asking, is there a mailing list that people can sign up for to learn more about the research going on at the bird collections?
So I tell you to follow the Birds Facebook page, where you'll see, it's mainly John and me and Mary Hennon who do it, but if you look back through that, you'll see hints to the research and collections that go on. The DNA Discovery Center has a Facebook page that I also run, and you'll see broader perspectives of the research that's on there as well. We don't have a newsletter. Yeah.
Great, sounds good. So I guess we can open it up. If anyone has any specific questions. Judy Pollock just asked one. Edward, do we want to have her ask it?
Yeah, Judy, if you want to go ahead and unmute yourself and ask your question directly.
Yeah, that was a great presentation. I was just wondering, you said something like birds look unique, but what they share inside is complicated. And I was, thinking about the relationship between like the structure of a bird, the morphology, and what you've learned from DNA. Like, do you, just going back again, do you understand better now about how birds evolved? Like you gave the example of swan, but what are some other things that you've learned? Like whereas if you were to look at a bunch of birds now and have to group them because of what you've learned from the DNA, you'd approach it differently.
I mean, I think that, if I laid out all of our specimens, you would have no problem figuring out which bird was a parrot, for example. That would not be complicated, and it's never really been that complicated. So a lot of birds kind of are like that. They share characteristics that let you bin them, but that doesn't let you understand the relationships. And I'd say it's a iterative process that you need both. You need to understand the morphology in detail, and sometimes understanding the relationships can kind of clue you into things about their biology and their morphology that you might not have seen before. So, that's certainly the case of looking at pathways of vocal learning. And, John can talk more about some of these things, but you need both. And then at his level of taxonomic hierarchy, songs, the actual songs themselves are really important to understand too, the genetic basis of them. So I think it's all iterative. No one is better than the other. The reality is that even whole genomes can be really hard to analyze, and there's all kinds of weird things that can happen in genomes too. Selection's powerful.
Great, thank you. Next question comes from Kat O, if you'd like to unmute yourself.
Hi there, I was just wondering about the timing of when did you run these analyses? How recent are they for the various families that you were talking about?
So the initial first publications were 10 years ago, and then subsequently we've done more analyses with larger taxon sample sets that have come out since then. Other people have done whole genomes, but on very small numbers. So that's one of the things that's really important to understand, is that tree formed a framework that then guided people who could do larger and larger analyses. And now, we meant that kind of like that people would use that to leaf the rest of the tree out, which is what's really happened over the last 10 years, of those relationships, as that's the structure of the tree. And now everybody's leafing out all the branches, which is an analogy I really like. And I think it's really great that science only gets you more questions than it ever gives you answers. I could never answer all the questions that were generated by this. And if I could, all the technologies would change, and I could ask different questions, or I could ask the same questions with different tools, which give you different insights, which I think all of these studies are actually doing.
So Kat, one thing I was going to mention is that when they first did this study, they weren't comfortable with the models to try to estimate the actual ages of divergences, so that there was no time associated with that tree. There was just a branching pattern. And now that's something that's also been filled in, and dating molecular trees has a bunch of assumptions with it. But at the same time, we're getting a much better idea of how old and when those lineages actually began to diversify. And
one of the important things about that is that the taxonomy doesn't tell you that. So we did not sample our species based on taxonomy. We sampled them based on DNA hybridization study that had come out that told us what the deep lineages of birds actually were, even though some of the relationships were kind of suspect at that. But the degree of genetic divergence is what formed a major lineage. So in our study, songbirds were undersampled compared to their taxonomic representation because they're one of the more recent groups of birds. And so a few of those could satisfy for all of the 5,000 of them. But we had some of the lineages deeper in the tree that had maybe one or just a few species were included because they were such genetically distinct lineages that they encompassed a lot of the genetic depth in the tree.
Very cool. Next question comes from Mackenzie Roof, if you'd like to unmute yourself.
Hi, from my very limited knowledge on evolution, I was wondering if the similarities between birds that were once categorized together that now we understand are actually unrelated and they share like similar morphology, if it's actually referred to like as an example of convergent evolution.
Yeah. John, you should talk about shrikes.
What about shrikes?
About the fact that things that can be placed in the same tray for 150 years are now different, and they actually aren't each other's closest relatives.
Yeah, so I mean, so the interesting thing about that is it ends up working both ways. So in some cases, that's exactly what's going on. We realize that certain sets of relationships are the results of convergence, and it can be on a trait by trait basis too. So for instance,
do you have long claws, and do you have that picture? I don't have that picture.
Yeah, I don't have the long claws, but for instance, gulls have webbed feet, and so do ducks, and yet there were enough other characters that nobody placed the gulls and the ducks together historically, and they still don't go together. I was thinking of this, the falcons are a perfect example of where everybody looked at the bills and the claws and the behavior of falcons, and they said they're belong with the other birds of prey, and that was not questioned at all.
But to me, that's not convergence.
Not necessarily. So some parts of it are. There we go.
Yeah. Now that's convergence.
Oh,
so that's a meadowlark and a long claw, and they look very similar, as most grassland birds tend to, with the yellow and the bibbing and the streaks. But those things are not each other's closest. They're not even closely related at all. And so that's an example of convergent. Thanks, Nathan. That's an example of convergent evolution. Whereas something like the hawks and the owls and the falcons, I think is more a lack of evolution. So that's a retained primitive characteristic in that lineage. And I don't think it's convergent. I think it just didn't change. But now that we know that, we can ask, well, what the heck happened to make parrots so freaking different from parrots, and then to make songbirds go absolutely crazy? Why did they speciate so much compared to some of these other groups? In the context now, we can ask that question and we can get some answers that are different than we would get without the tree. So does that help?
Yeah, definitely. Thank you.
So we had a question from Fran. If Fran would like to unmute himself.
He actually is in walking distance of our house, probably.
Can wave from each other down the block.
Exactly.
Hey there. First, I have to say I've been on the edge of my seat tonight. This has been really a tremendous presentation. Shannon, we met years ago. We
Right.
have mutual friends, the Fens and the Hevicks, and it's been a while since I've seen you. I do Bird with John occasionally, but this was awesome. Thank you. So there was a slide earlier that mentioned something about field guides, almost like, what do we do? And I'm curious, is there anything that could change or should change with field guides? And is there pressure from publish houses or authors? Like, who's pushing back on making some of these changes to better depict the reality of what's been learned lately?
Well, I would push back if I were a field guide publisher too, because I'd be like, what the heck? You want me to change every single thing? You want me to move the a lot of plates were painted without an understanding of the relationships of those birds. And John and I have both watched John O'Neill do this. And he's a really famous bird artist. And we knew him at LSU, and he didn't like the way some of the pictures and some of his plates were, and he actually cut them out of the plate. And I thought my heart was going to stop. He just like took a knife and cut the bird out of the middle of the plate. You're like, what the heck? But you realize that that's what would have to happen if we were going to make field guides represent actual relationships. And so if I had my way, one of the things that would happen is that there would be trees in the field guides, like actual phylogenetic trees showcasing the relationships of these species, because then you can, tree thinking is really important. If you know nothing about trees, COVID-19 and the SARS-CoV-2 virus itself should tell you how critically important trees of life are and this kind of variation is. And so I guess in the context of people dying, the tree of life for birds seems less, less serious, but it is a really serious thing for people to do, to understand, because you never know where insight's going to come from, where people, they'll look at the world in a different way when you tell them what the relationships really are. So it's easier now. Things are digital. You can move things around easier than you could 20 years ago or 30 years ago when I first started doing this stuff. So I don't know. And field guides are changing. There is a much better taxonomy section in the beginning of most field guides. It's way better than it was, because of the recognition that what we know now about the tree of life means that a lot of the things we thought just aren't true. I mean, flamingos and grebes are each other's closest relatives. There's not a field guide in the world that would have told you anything like that until relatively recently, because you look at them, you're like, what the heck, no way. But it's true. So I don't know. People who are really creative are going to have to come up with ways of figuring that one out. Does it matter to you? But does it matter? It might not matter to people the way it matters to me.
I think go ahead, Fran.
No, I think it matters to birders and people wanting to learn, and textbooks and all of that. But so it'd be nice to know, have a reference. And I'm glad you said some of them are changing, because I was going to say, like the Merlin app or any of these that are more digitized, are they more reliable maybe? And
it's easier for them to be reliable, right? Yeah. Because, they don't have to do nearly as much work to reflect what is modern taxonomy. And, John and I have these disagreements all the time too, but I actually think there's 100,000 genetically distinct lineages of birds, which will drive most bird watchers absolutely crazy. And many of them you cannot tell by looking at them what they are. You need to either sequence them, listen to their songs, or know where you are in the world. So, that's a really complicating thing too. What is a species? I actually don't believe in species, but John does.
But what I was going to say, Fran, is that I've been fascinated. So most of the apps are designed to help you identify things, and so they're not coming at it from the perspective of the evolutionary side. And so, from that perspective, they would never put flamingos next to grebes necessarily. You'd want to be able to identify things that you're seeing in the same place. And so, the neat thing about apps is that you have the potential to put in both and make it work. And I don't think there is anything out there that's done that yet.
And I guess one thing I would like to, ask you both is, all of the things we've been talking about today have been enabled by collections and specimens and actually going into the field to get hard evidence and physical versions of these things we're talking about. And we live in an age of multimedia, so you often hear conversations surrounding this topic of, it's taboo now to collect birds and kill things. We want things to be able to, live without human intervention and whatnot. But would either of you be able to touch on the value of continued research, even in the era of wanting to photo document rather than have a physical version?
Yeah, what can a photo tell you about isotopes? What can a photo tell you about digestion? What can a photo, you know, a photo can tell you something about molt, but you can actually see the molt in a specimen. And so, you know, what I always like to point out to people is that it's not an either or thing, right? I've got no problem with people taking photographs. I actually think we actually have the archives of the Illinois Rare Birds Committee sitting in the museum. And I think that's a good thing because they're records of birds. But side by side with specimens, those specimens can be used for things that the photographs can't be. And so, you know, they both have their purpose. And one of the things I think is really phenomenal is, you know, the efforts that are going in places like eBird to make those records more archivable. But again, there's so many things you can do with specimens that you can't do with photos, that we really need to continue to monitor the world this way going forward.
So we can give you, I can give you lots of bird examples, but a topical example now has to do with the bats that people think are potentially the jumping point for coronavirus to get to humans. We know very little, apparently. This is not my area of study, but the mammal curators tell us we know almost nothing about the relationships of those bats. They're full of undescribed taxa that you know, have fairly unknown distributions at this point, and it's only through rigorous analysis of specimens in museums and rigorous trees of life that we'll know which population of the bat might be contributing to that. And it's only through collections that we can go back through the collections and maybe look for evidence of this viruses in our tissue samples and in our specimens. So you know, a lot of virus hunters go all over the world, and I'd put Dave Willard, for example's ability to identify a bird against virtually anybody in the world. Certainly if a feather, I don't think there's probably a person alive right now who can identify bird feathers better than Dave. But the reality is that we're all fooled. Ted Parker was fooled by bird songs even at times. And we can't identify things in the field with enough reliability for things that are critical. There's tons of undescribed variation. You know, and it's only once you have all of these things together that you can put together that, oh, the behavior that bird did actually is diagnostic of a different species. It's only after we put the tree of life together for Darwin's finches that we could see that what defined Darwin's finches and their relatives is the way they build a nest, that they build dome nests with side entrances. All the birds in that group, all the birds in that clade, they all do that. And that is a defining feature of that relationship, but we didn't know it until we had the tree with all the specimens and the description of the natural history. And all of a sudden those things they emerge from our knowledge of the specimens, and there's nothing else that can get you that information. And birds are changing. John Student, the collision monitors, Dave Willard's, the study of specimens over the past 40 years, which when John and I got here was called undirected collecting, and people looked their nose down at us. I firmly believe that every living thing should die by the museum, and it should all get turned into a study specimen so that it's immortal when that happens, right? You know, including our son's baby teeth, which he's not on this call. So we collected our son's baby teeth. They're in a container in the museum because there was no baby teeth from a human in our museum. But there's nothing that can substitute for that. No human has enough knowledge. No human can identify these things. Even with advanced study, we have trouble identifying things. We make mistakes all the time, and we hope people can come after us and correct mistakes. They can only do that if they have the physical things to hold on to and to study. We didn't know birds saw in ultraviolet wavelengths 20 years ago. Now we do, and now spectrophotometry comes along and enables us to see, well, we can't see it, but we can infer all of the patterns and colors that are in birds that they can see, because we now know that they have extra sets of cones that we don't have. And so I can go back and then we can look at specimens from 250 years ago at the beginning of our collections, at the beginning of collections, and we can see things that we could never see with new technologies. So that's what dying gets you when you're an ornithologist. You don't get to see what comes next. But the thing I know with 100%, it's one of the only things I know with 100% certainty, which is that if we keep these collections, that they will form the basis of science forever into the future, with ever new technologies enabling us to look at new ways at even old specimens.
Very, very fascinating. The final question that we have came from Katie Craigbaum. She's asking, what is the what branch of the bird tree still requires a lot more so-called leafing out?
Asian birds.
Yeah.
In my opinion,
Yeah.
well, if there are British people on this, you know, you'll have to forgive me a bit, but anywhere the Brits have been for a really long time, there's a lack of, there's been a lot of study of birds and behavior, but there's an actual lack of the taxonomic understanding of birds because there aren't enough specimens, and there have been no modern specimens that have been collected from a lot of these places. And so the only thing we know is that every time we study things, there's things nested within species. There are populations within one species that are actually more closely related to other species. I'm not sure what a genus means, because there's always something that goes in that we didn't know, and always something that comes out and goes somewhere else that we didn't know. But I would say the biggest areas are Asian birds, and that includes African birds are way better understood now, I think, even than Asian birds. What do you think, John?
Well, I mean, there's been some, I agree that there's a ton of work to be done in Asia, in part because of all the isolated populations on islands and things like that. And Shannon's right that it hasn't been studied in detail for a long time. But, you know, again, with the new tools of genetic work, there are things out there like tapaculos, for which there's now work in the Andes showing that you know, Shannon's argument that there are a lot more lineages out there than we realize are starting to show up in these studies, where they're describing that. I'm trying to remember the numbers on that genus,
I think it was 30 new species they need to describe, right? Isn't that right?
Within the last, you know, and so again, you know, the example there is those things are morphologically and behaviorally almost like mice, and they have very small ranges and have a high propensity to once they expand into the next river valley, develop an isolated population. And so we've got some good ideas on where to look for things, but another, you know, there are surprises too. So another interesting example is Prosolariformes. You know, people didn't really think about the biology of Prosolariformes, but there's strong.
What's a Prosolariform?
Sorry, petrels and shearwaters for the most part, but also albatrosses. And, you know, what they weren't paying attention to with respect to these birds is that they go out to the ocean and they go for thousands of kilometers, but then they have to come back to breed and find a mate. And so there's very high selection pressure to come back to the very same island where you came from. And as long as you do that consistently, you're going to start differentiating from another population of something that you're at one point part of the same thing. So the number of Pterodroma petrels, for instance, has skyrocketed because people have realized that every one of these islands actually has a genetically distinct population, and then they're starting to find the subtle morphological characters to differentiate them too.
Very cool. And I guess the final question before we wrap up that I would just love to know is there a good place for people to be able to go to sort of learn about the concept of a species or taxonomy? Like, it's a daunting question.
No.
And there's no textbooks either. So there's no textbooks on systematics. There's no, I mean, there are some good pieces, but of literature that are in the scientific realm that people still fight about. I mean, I had to joke because someone just had a dissertation defense when they talked about their committee talking about what a species is. And then Jason Weckstein, who is also a student of Bob Zink's, talked about that exact same thing. And then I chimed in all on Facebook that's what every single one of my committee meetings was about, a fight about what a species is. We had a fight just a couple weeks ago on John and our lab group discussion about what a species is. That's a never-ending argument that I don't think can be won because I don't think species are real.
Mm-hmm.
Well, and on that lovely
Katie.
note, that species aren't real.
I know,
can we
that
get a
sounds,
huge
that
round of
sounds too
applause
negative.
for
but-
Shannon and John? Thank you guys so much. Please hit your virtual emoji applause buttons down there. But thank you guys so very much for joining us. This has been a really interesting topic. I know some of us are coming out of this with our minds kind of blown here and not really sure what to do with all of this information, but thank you again so much for taking this evening, spending your time educating us, and sharing your intense depth of knowledge. So thank you so very much.
We're really grateful.
Again, thank you out to On Tour Brewing for also being a part of the stream with us. If you've got a chance, definitely consider supporting them and for your next booze needs, supporting them because they're great supporters of the bird community. We'd also like to give a quick shout out to Chicago Audubon and Peking Notabar Nature Museum, who are also partners in these Birds and Bytes series. And then thank you to all of you guys who joined us here this evening. We really appreciate it. We appreciate you guys again taking the time out of your evening to join us here, learn about birds, have a good time, have a drink. And if you haven't already, check out our website at chicagobirder.org, where our next couple of Birds and Bytes are posted. And hopefully we'll see you guys again next time. Thank you guys so much. Have a great evening.
Thank you all. Have a good one.